Okay, thank you for being here today. My name is Mark Williams, and I am a professor at the University of Kentucky in the Department of Horticulture. I'm also the director of the University of Kentucky Organic Farming Unit, and I've been involved with oak at various levels since its inception. But it's been a few years since I've been here, and it is so nice to see so many new faces and young people. So great to be here today. This session is going to focus on, I think, one of the most important components of our agro ecosystems, and that is the microbial community. We're going to take kind of a full spectrum approach and talk to you about what science can tell us about the way that we manage microbes, the way that these microbes in the soil, the way that these microbes can then impact plant health, and the way that plants can impact the human microbiome, even maybe from a microbial standpoint from what's inside the plants. And so we're going to have three speakers, all experts in their fields. Our first speaker will be Dr. Shawn Lucas, and he's going to talk to us about what we know about soil management and how it impacts microbial community health. For a long time, we've known that farming practice impacts the microbial community, and even for the last couple of decades, there's an increasing amount of evidence that organic practices compared to conventional practices increase things like microbial community diversity, abundance, things like that. So we're going to hear more about that. And then Dr. Carlos Rodríguez López, who is an expert in plant microbiome interactions, is going to show us how microbes impact plant health at a genetic level even, and how that can impact plants' abilities to interact with their environments. And then lastly, Dr. Kendall Corbin, who's an expert on human microbiome, will talk to us about what we know so far about the way that what we eat impacts the human microbiome, and even maybe from a microbial standpoint. And what we're going to see in this kind of spectrum approach, or this continuum approach, is also like a spectrum of what we know scientifically. So there's a lot known about the soil, all the way to what's right at the cutting edge of what we know right now scientifically. So the goal of this session is for you to have a deeper understanding of how farming practice is not just impacting the soil when you're out in your field thinking about some of the core principles of organic agriculture, like feed the soil, let the soil feed the crops, but how what you do is actually impacted all the way through to your own health through these microbially mediated processes. And so our first speaker is going to be Dr. Lucas. Ready to take it away?
I'll take the high ground. All right. Thank you, Mark. And it's good to be at Oak. It's always good to be at Oak. This is usually one of my favorite. It is my favorite conference every year, I think. And I can remember when I first came to Kentucky, I was working with Dr. Williams. He was my advisor, and we used to have really cool conversations. When we had more time, we could meet in Mark's office and talk about some of these real, what we considered esoteric ideas about, hey, it would be real cool if you could think about the connectedness between what we do to the soil and what we do to the microorganisms in the soil and how we impact them with our management, and how that in turn translates to changes in plant microbiomes, and ultimately maybe even our own microbiome in our body. And this was, 15 years ago now, at least. And so the field has grown and it has changed, and I'm excited to be part of this workshop, and I guess I'll just go ahead and jump in and talk a little bit about some soil basics and soil microorganism basics. Maybe. There we go. No, I see my slide here, but not there. I think it's coming. There we go. I teach here at Kentucky State, and so I'm just slip into teaching mode for a minute. What's a microbiome? Anybody want to give me a shot at defining a microbiome? Yeah, okay. So a microscopic community. Where can we find a microbiome? Awesome. So, all right, so that gives me a good starting point. There's the human microbiome, the soil microbiome, the corn plant or broccoli plant microbiome. I'm going to focus a lot on soil, and Carlos and Kendall are going to take it up from there, right? Soil is the foundation of everything, and so I'm excited to jump in. Oh, wrong way. Going the wrong way already. When we think about soil, it's complex. It's a very complex environment. You've got sand, silt, and clay. Those are our mineral components. Then you've got organic matter, which if you've been at the Oak Conference long enough, you can't escape talking about organic matter at some point, right? But then you've got these little creatures that live in the soil that facilitate a lot of these different processes that we take for granted. And so I just wanted to give us sort of a basic overview of who's down there, who's doing the work, and so we can think about bacteria. These are little single-celled creatures. The scientific word is prokaryotes, but they're just single-celled creatures, and they carry out all kinds of different functions, from helping plants do nitrogen fixation to decomposition processes, nitrification, denitrification. They're involved in all kinds of nutrient transformations. And then, so again, these are things that, as people interested in growing plants, to have a little bit of an understanding of what they're doing, that's helpful, right? So this is a cool picture, and I tried to make this picture heavy. This is a scanning electron micrograph, so a real powerful microscope, and it's showing us different kinds of bacteria. Where's my pointer? Yeah, there we go. I don't know how well that works. You can see different shapes, right? You got rod-shaped bacteria and sort of spherical bacteria, all kinds of different organisms in there. This is on the surface of a clay molecule, or a clay particle, rather. And then we think about fungus, right? Fungus. And the cool thing to me is when we think about bacteria and fungus, what do we usually think about? Yucky stuff, right? Diseases and stuff impacting us negatively, right? And of course, we've just been through this pandemic thing, and microorganisms are, oh my gosh, we want hand sanitizer, but really most of the microorganisms that we interact with on a daily basis, they don't do anything bad to us, or they help us, right? And so it's important to keep that in mind. So fungi, yeah, some of them cause athletes' feet, but a lot of them, live in the soil and do good things, and so they're basically filamentous creatures. They kind of look like roots when they're in the soil, but they're smaller than a plant root. The picture on the, all right, get my pointer right. This is basically a decomposer, a wood rot fungus, specialized in breaking down wood and other organic material. And here's a mushroom, right? When we think about sort of fungi in the forest, very often we think about mushrooms. Mushrooms are just part of the fungus. They're the reproductive organ, essentially, of a lot of fungi. And then this guy up here, I love this picture. I show this in some of the classes I teach. It's one of my favorite kind of soil geek pictures. It's a mycorrhizal fungus that lives both on the root and inside the root. And so that bigger structure there, and again, my pointer doesn't have the resolution I'd like it to have, but that's okay. The sort of the thicker tubular structure in the middle of the circle there is the tip of a plant root. And all those little filamentous, wispy little things coming out of it are this mycorrhizal fungi. And the fungi get into places where that plant root can't get and help that root get nutrients, right? So these organisms really do facilitate plants getting nutrients. I've already gone down to the 10-minute mark here, so I'm going to speed up. Actinomycetes are bacteria that kind of look and behave like fungi. Nematodes, they're worms, but they're not earthworms. Non-segmented round-worms that carry out different functions. Some of them cause disease. There's root rot nematode, or root knot nematode, but other nematodes, feed on bacteria and fungi. Remember, this is an ecosystem in the soil. It's a complex ecosystem. Lots of different organisms. So this is just some scientific facts. Some of them might be a little out of date. They're a few years old. But just basic estimates. So 150,000 different bacterial species in a tablespoon of soil. 522,000 actinomycete cells per tablespoon of soil. 15 different mycorrhizal fungal species per tablespoon. And in a typical forest, you might have 400 or some different species of fungi, right? And keep in mind, a forest soil is usually more complex than an agricultural soil. So, it's not always a straight translation from some of these to what we see in our agricultural fields. And if we've mismanaged our soils, some of these numbers might be lower. And again, I said this is an ecosystem. So there's all kinds of cool interactions going on. So up here we have interaction between bacteria and plant roots. So the bacteria are living, this is hairy vetch. I grew this a few years back. Thought it was a cool picture. You can see the nodules on the hairy vetch root, and that's where the bacteria are living and doing nitrogen fixation. Here's a similar picture to the one I showed before, another cool picture with some mycorrhiza, but also with some decomposer fungi in the same picture. But again, all kinds of interactions. That decomposer is releasing nutrients into the soil that the plant root might be able to get, and then the mycorrhiza are able to help, they facilitate that process of nutrient acquisition. And in this picture, I just love this picture up top, all right? So this is a fungus. So some of these nematodes like to eat fungi, but sometimes the fungi like to eat the nematode. And so this is a nematode, and he swam through the wrong little area and got caught in some of these, this is an electron microscope picture. He got caught in some of these little balloon-like loop structures, and when the nematode swims through the structure, the balloon inflates and captures it, and then the poor guy gets digested while he hangs out there, and the fungus takes up its nutrients. So sometimes you win them, sometimes you lose them. I should go back. I wanted to point out, soil is an extreme environment, right? Either there's, residues on the soil or in the soil that the microorganisms can chew up, or it's like a famine, right? It's feast or famine. Depending on the soil, there can be different extremes in pH, moisture, wet and dry cycles, and the wet and dry cycles affect the amount of air, so you can have microorganisms that do better when there's more air and microorganisms that do better when there's anaerobic conditions. And temperature. All these things are going to affect who is active in the soil at any given time. You may have seen this one before, but this is kind of showing the different interactions. The whole system is driven by photosynthesis, right? The photosynthesis starts with the sun, and the plants capture the sun energy, transfer some of that energy into the soil. As they decompose, the microorganisms decompose the plant debris. And then you've got other organisms, fungi and nematodes that eat on the bacteria and the smaller fungi. And we could keep going up, to the different levels of what's essentially a food chain, right? But eventually all this stuff dies, right? And so then it goes back through the soil and decomposes, and it facilitates nutrients getting back into these plants. So it's a big cycle, right? Driven essentially by the sun. So microbes are important. I mentioned a few of these already. Decomposition, nitrogen fixation, availability of nutrients, biological control, diseases and pests, plant growth promotion. Some microorganisms release hormones that can stimulate plants to grow in certain ways. Some microorganisms impact drought tolerance. Soil structure. I'm a soil scientist by training, and so I think a lot about air movement, water movement in soil, and the bacteria and the fungi can influence that. And so when I got to UK, one of the things that Mark and I often talked about is, can we think about managing these microorganisms to do specific things? And so, we came up with a project that we were thinking about, can we use different amendments to promote structure in the soil? And again, structure impacts water movement, air holding, root penetration, nutrient acquisition, all these things that we want for our plants. We had a couple different soils. We beat them up really bad, destroyed the structure. We incubated them for 82 days with either vetch, compost, or manure. And then we examined like the formation of aggregates, which is basically the building block of soil structure. And we also looked at relationships between that aggregate formation and microbial activity, essentially. And so between the three amendments that we used, and again, we beat these soils up. We passed them through a very fine sieve so that there was essentially no structure. When we added hairy vetch to the soil, it promoted the most aggregation. Manure was kind of the second most. And then compost, and then we had a control where there wasn't anything, didn't really do much, right? Compost didn't do a whole lot compared to the control. Did a little bit better in some soils, but not all soils. But the ones that had more organic material that was easily decomposable, because compost has already been decomposed once, right? The microorganisms have basically chewed it up. There's not a whole lot of active material there for them to re-eat. But where we had vetch and manure, there's a lot of readily usable carbon there that they could attack, and so that microbial activity kind of stimulated this aggregate formation. And these two photos, take my word for it, I don't have time to go in depth on it because Linnell's going to flash a number at me here in a minute and tell me I'm out of time. But these two graphs basically are kind of showing us that the manure and the vetch stimulated the most fungal productivity in 3 different soils. And ultimately, we were able to do some analysis that showed that there was a pretty strong relationship to where we had more fungal growth. We also had higher levels of aggregation, right? So kind of taking the whole thing and tying all the different pieces together. And you could actually see it. The third column in C has fungi growing in these, on the surface of the soil in these containers that we incubated. So it's kind of cool to see the results that you see in the lab from instruments and lab analysis. It's practically observable in containers, right? So basically, we came out of this and came out with different conclusions. We could sort of drive the microbial community to do a certain thing, promote fungal growth, and stimulate aggregate or soil structure. I'm going to skip some of these because I want to get to where we're going, right? I did a little homework tonight or last night, so I could talk about this today. And people are looking at these systems now and thinking about how do we manage these? We want to be tailored, we want to be predictive, and we want to be integrative. And this paper that I read out of Nature is pretty cool. It was talking about how we could potentially be real specific about how we think about using microorganisms to promote certain outcomes in agrological ecosystems, but it ties in with the host, right? The host being the plant or maybe the bees. And this blew my mind last night because we had all these conversations about, the soil, the plant, and the human microbe, but insects, right? Insects are part of this equation too. And I, as a soil geek, I had never really considered that until last night. I'm like, well, yeah, that makes sense, right? The flower, the bees visit the flower, and their microbiomes could interact too. And so the host could be the plant, or it could be the insect, or the host could be the human microbiome, right? And so tailoring management of microbes to have certain outcomes, beneficial hopefully, right, involves management practices. What are we doing with the soil? But maybe there are certain plant types that exude substances into the soil that promote more microbial activity. Certain cultivars might do better than others. And then the last part is like people trying to really get reductionist on it and boil it down, right? Almost like giving a specific pill for a certain problem, right? And I don't necessarily know if I like that component of it, right? So the take-home points, and I just got the hook here from Linnell, so I'm going to finish this. Think about what, what's out of sight. We take what we're doing for granted sometimes in some ways to the soil. The way we manage soils can influence the soil microorganisms, and if you're an organic farmer, those soil microorganisms are the key component in nutrient cycling and these processes that we depend on so much to produce the crops that we want to produce. Think about how we might be able to control them, but really, we're all organic in here, or most of us are thinking organic. Sometimes it's just, you don't have to go too deep and think about being so highly tailored the way that this article I read suggested. Just keep doing the good soil management practices that you're doing, and you're having some positive impact. I think there's some evidence for that. Building soil organic matter is the key. I'm a soil scientist. I've studied soil carbon for 20 years. It's cool to see everybody thinking about it now. That's the key to all of it. That's all I got.
Okay, hello everyone. Can you hear me? Yep. Okay, well, I do not have a title slide because really this is a continuum. There is no need to say, here's where I start. This is just a continuation of what Shawn just told us. As he mentioned, the soil microbiome produces or gives the plant a lot of good things, including better access to nutrients and water, protection against pathogens, as Shawn mentioned at the end. It also attracts beneficial animals like nematodes. How was this? Oh, there you are. There you are. Nematodes and pollinators. Yeah, I have to look there. This is weird. And parasitoids that protect plants. And well, we all know what happens if we didn't have pollinators, right? We wouldn't have many of the fruits that we consume. But it's not only that. A few years ago, we were studying the potential effect of the presence of specific microbes associated to plants and their contribution of the quality of our produce. This is correlations that we found between fruit and wine quality in grapes associated to the presence of a specific bacterial associated with the plant. So now we're starting to understand that a lot of the concept of terroir is associated to the soil microbiome and how they interact with plants and affect the plant production of chemicals. So I have to admit that I steal this slide from Shawn. Thank you very much. And what I'm going to do today, I told Mark that I was going to talk about something else, and then as I was preparing my slides, I realized that I really wanted to talk about something different. So I think what I'm going to do today is tell you what is it that the work that I do associated with plant and microbiome associations. Okay, so as Shawn mentioned, there are different approaches to use these beneficial associations between plant and microbes, and what I really want to understand is what are the genetic component of the plant controlling the association between the plant and in this case the host and the microbiome. So that's the part that I'm interested on all this part. Some people say that this is really not possible because it's such a complex interaction that there are so many factors affecting this relationship that we really cannot. The last idea or the ultimate aim is producing or breeding crops that are better hosts for the microbiome and that can utilize those beneficial benefits from the microbes. So, in reality, we've done this already. So through domestication, we've altered the ability of plants, of crops, to associate with microbes. I don't know if you're familiar with this crop. Anyone? Well, it's written there anyway. This is NSAID. It's a crop that was domesticated relatively recently, around 6,000 years ago. It's originally from Eastern Africa, and is one of the most important crops in Ethiopia. Some people call it the tree against hunger, but you might know it as the false banana. As you can see, it's very similar to the banana tree, okay? This is a very big grass, let's put it this way. So, A few years ago, I had a student from Ethiopia, and he was interested in studying how domestication in his country, in Ethiopia, affected the genome of NSAID. So he went back to Ethiopia, collected around 250 genotypes or different plants from this crop, both cultivated and wild accessions. And here, what we did is use next generation sequencing to sequence the genome of these plants, and we compare those domestic accessions to the wild ones. And as you can see, basically their genomes are very different. For those of you who are not familiar on how interpreting these graphs, we take information from the whole genome. This is 6,000 SNPs across the whole genome. And if you've got each of those circles is one sample, and if two circles are very close, their genomes are very similar. If they are far apart, they are more dissimilar, okay? So as you can see, the reds are quite different from the wilds. So this is what domestication has done. We have selected a specific set of alleles, or let's put it that way, from the total population, and we have end up with something that looks very similar, but genetically is quite different. So we did some reusing. So this is almost organic science. We reuse this data because every time that we sequence the genome of something, at the same time, we're sequencing the microbiome associated to that organism. So what we did was, okay, let's remove everything that is from any sequence that is from the plant, and let's see if we find sequences from microbes in that information. And this is what we found. We found that the microbiomes of domestic and wild NSAID is very different, okay? So this has been shown in many crops, but this is particularly interesting because, as I mentioned, this is a crop that was domesticated very recently, 6,000 years ago, compared to rice, for example, that it was domesticated around 13,000 years ago, okay? So if we've done it in one way, I think that we can do it again on the other way. Because what we've done with domestication, and this is for many other traits in plants, is the same. We've altered the ability of crops to associate with their microbiomes. In a way, we've made them a little bit stupid. And that's the reason why if you plant a crop and you leave it on its own, it's probably not going to succeed. But if you take a plant that grows naturally in an area and just drop a seed there, it's probably going to survive, okay? So we need to take care of crops in a way because through domestication, we've made them a little bit stupid. Okay, so there is one problem. As I mentioned before, there are many, many factors affecting how plants associate with their microbes, okay? So anyone cares to tell us what do you think is the main contributing factor to the microbiome composition of a plant, of a given plant? Anyone? Management? Okay, close, but not cigar. Anyone? Access to? Definitely. If you don't, you cannot associate to a microbiome, and this is a specific bacterial or fungal species if it's not there, okay? So that's definitely a really important factor, and that's associated to the environment, okay? So the most important factor affecting the microbiome composition of a given plant is the environment. So let's see an example of this with crops. This is a study that we did taking data. Again, this is reusing data. This is not data that we generated in my lab. What we did was to go to publish articles, and we collected their data. This was a microbiome data from 885 vineyards across the whole world. And in that data, we were able to find 80,000 different species of bacteria alone, okay? So it's a lot of data, and what we found is that the microbiome of those vineyards group according to where it was planted. So in this case, I'm showing it here by country, just for simplicity. So telling us that the main factor controlling microbiome diversity is where the plant is planted, basically, okay? So the next, in a cultivated area, the next factor controlling or affecting diversity is management. You've probably already know that the use of organic versus conventional agriculture, or till versus no till agriculture. Some people say that till agriculture doesn't affect the soil microbiome. I don't think so. But also what sort of chemical inputs you're using, okay? But it's not only that. This is also some of the research that we've done. We found that how close you plant, so the space between your rows and the space between your plants is going to affect the microbial communities in the soil, okay? So silly things like that really have an effect on the soil microbiome. Okay. Two other factors that many people do not take into consideration is that the plant at different ages or different developmental stages have different needs. And they're going to associate with different type of microbes depending on where they are in the life cycle, okay? This is an example using the evil soybean. And as we can see in this graph here, okay, so on the vertical axis, there we've got a measurement of how different is the soil microbiome community associated to the plant compared to the original soil microbiome community without the plant. So as you can see, as the plant grows, it starts to alter the soil microbiome, and it gets more and more different. But at some point, and this is at R6, R5, that's when the plants start to fill the seeds. At that point, the plant says, okay, I don't care about myself anymore, I just care about my seeds. That's how the plant is going to survive next year, okay? So it starts investing and putting an effort on controlling the soil microbiome, and the soil microbiome will revert to the original eventually, okay? So the association between the plant and the soil microbiome is temporal. We have to remember that, and in a way, it's selfish. There is an association, everybody benefits, but until the plant says enough is enough, okay? So, propagation system. I think I forgot to put an extra slide here, but another factor that affects the microbiome associated to a plant is what's the origin of your propagule. So you're not going to have the same microbiome on a plant propagated from a seed than from a cutting. And that's due to epigenetic mechanisms regulating that association. Sorry. And finally, the cultivar. So you think that maybe that what we think is that small differences between cultivars have an effect on the microbiome that the plant is capable to associate with. So going back to that study that I was showing you before, okay, here. We found that when you compare the microbiomes of vineyards across the whole globe, the whole world, they're significantly different from each other, depending on where they're growing. But what we did was to use artificial intelligence, and we trained an algorithm to see if we could predict what was the cultivar of grapevine that was planted in those vineyards, okay? We had 34 different cultivars, and what we found is that, yes, the algorithm, the artificial intelligence, was capable of telling us what was the cultivar planted there, just looking at the microbiome. So the way to interpret this graph is that what you want is a dark rectangle in the middle. So if you've got a 1 or a very dark blue, that means that the algorithm predicted correctly 100% of the times what was a cultivar planted in that vineyard, okay? So as you can see, the majority of the times, the algorithm was capable of telling us, well, this is Cabernet Sauvignon planted in Spain, just looking at the microbiome. What this is telling us is that the genotype of the plant, there are very small differences between these genotypes, is controlling really in a really strong way what is the microbiome that is associating with. And I think I'm going to stop there because I'm out of time. Thank you.
Okay, so I'm Kendall Corbin. I'm an assistant professor at UK in the Department of Horticulture. And so Mark said we were all experts in our field, but I feel really inclined to be honest with you guys and tell you that I'm actually the butt of many jokes. I'm a shit scientist. And I mean that quite literally. So when I say that my research program is really interested in bridging the gap between agriculture and human health, one of the tools that we use to do that is fecal samples. And so we can do research using your stool samples to try and get an understanding of what's happening in your gut, what are the microorganisms in there, what are they doing, and looking at really cool things like diet and gut health. And so maybe I'm a little traumatized, I'm not going to lie about it. How many people remember the food pyramid? Do you remember the rules? There's some people that do, 1%, okay. Did you follow it? It was incredibly impossible. How could you eat 5 apples, 27 bananas, no bread, no carbs, no sugar, okay? So I don't even know if they teach this in school anymore. Probably people with kids have a better idea. But I think our mentality about how we approach food and what does a balanced diet look like has drastically changed in a very short period of time. And so really, what is that missing link then? If you do eat 27 bananas, 42 apples, no meat, no sugar, no Pepsi, no Cheetos, why are you not guaranteed to be fit and healthy just like me? You can hire me to chop down all your logs if you want. And that's because it's not that simple. And I think we're starting to see that in the talks that have come before me, that there's a lot of things that come into play, and there's this really critical missing link there. And in this case, one of the main missing links in trying to understand this relationship between diet and our overall health is those microorganisms that are going to be found in your gut. And in particularly, not only who's there, but what are they doing? And so, why are microorganisms in your gut important? So they're really important in the soil, they're really important in plants, and they're also really important for our health. And we have microbes all over us and in us, but for now we're just going to talk about those ones in your gut. And so they play a key role in breaking down food that your body can't digest. So think about, you just had this fantastic lunch. Some of the things that you ate at lunch were broken down in your mouth. Some of them were broken down in your stomach. Some of those food components made it to the small intestine. But those things that went through that roller coaster ride of enzymes and pH made it to your large intestine. And you actually fed the microbes in your gut. So you're feeding for you plus about a gazillion other people. So good job. And then when you feed those microbes that are in your gut, they break down the food components and they convert them into things that are really good for us, so like metabolites and vitamins. But your gut's not Vegas. What happens in your gut doesn't stay in your gut. So those metabolites and those really good compounds that those microbes are making actually leave your gut and go to other parts of your body. And so they also help protect us about harmful pathogens and regulate our immune system. But, like you've learned today, there are just so many factors that come into play when we try to ask questions. What is going on in our gut? What makes a healthy gut? How can we modulate our guts to be healthy? And some of these factors we have a little bit of control over. So like what we eat, how much we exercise, stress is a hard one, and the use of antibiotics. And some things, at this point in our lives, we're really just not going to be able to change them. We're not going to get younger. None of us are probably Benjamin Button. We didn't pick our host genetics. We live where we live. But diet is one of the main factors that on a daily basis we can kind of think about a little bit more critically and could possibly change for the better. Okay, So why so many questions then? I've told you everything you need to know. I've told you microbes are important for, that connection and bridging that gap between diet and your health. I've told you why they're important. I've told you it's complex, but we can make some changes. So why don't we have the answers? Why don't we have just this magic pill we can take to magically make our guts happy and healthy? It's because we're not Mickey Mouse. Most of the data that has come out and has really looked at long-term studies on diet have been based in mouse models. And can you imagine if I came in the room today and I said, okay, these people up here, you're gonna be on high-fat diets for the rest of your life. And you people back there, you're gonna only eat watermelon. And you guys, you know what, you're gonna get the chow. We can't do that with humans. We can't look at diet really across our whole lifetime or even generational. But we have information for mice, so it's a good starting point, but it's not enough. Another reason why we really don't have all these answers and there's still this really big gap that we're trying to bridge is that a lot of the research is looking at food when it's on your plate. We're not thinking about food and diet when it's still in the field and we're growing our plants. We're thinking about what is the composition of that food and how has it been processed? But we really, really aren't thinking about it big picture. And I'm guilty of that as well in my research, so I'll be the first to admit that. And so, it goes without saying there's a lot of catchwords that you've probably heard in relation to gut health and gut microbiome and how to make your gut happy. And so some of those words are probably prebiotics and probiotics. And so prebiotics, this is a little bit where my research kind of lays a lot, is compounds in food that induce growth and activity of beneficial microorganisms. So think about, okay, how many people in here love starch? Me too. I'm a lover of starch. Beige is my favorite color. If it's starchy and it's deep fried, I'm there for it. But starch is a really big term actually because there are different types of starches. So there are types of starches that are rapidly digested, so that means they get broken down in your mouth pretty quickly. You have slowly digested starches, and those get broken down in your small intestine, so about that 4 hours it takes food to move through there. And then there are resistant starches, and resistant starches, once again, that fraction of the starch that kind of escapes everything, gets to your colon and feeds those bacteria. And so that's a type of prebiotic that you can find in different foods. And then there's probiotics. And so anybody here like kombucha or fermented foods? Okay, so you have had probiotics then, whether you knew it or not. So probiotics are actually living microorganisms. And so you can find those, like I mentioned, in fermented foods, but you can also buy them in cocktails. They're all over the shelves these days, and I'll kind of leave it there. But there's this really cool emerging area of research where I think we are really kind of coming to realize that we need to take a step back and stop thinking about just diet and food when it's on our plate, but actually asking questions like, can we use that plant microbiome to modulate the human gut microbiome? So can we make this connection? And so this brings back to really, I think, at the core of what my research program wants to do, and that's to really think about, if you want to have healthy plants and you want to have healthy people and you want to have a healthy planet, what's the link between all of these things? Microorganisms, exactly. And so if I went and got a stool sample from every single one of you all, we did some sequencing and looked at it, we would have some commonality. We would have some microbial signatures that, we're all gonna have. But each of our guts are going to be very unique and specialized for you. The same for soil. We go and get a soil sample in Appalachia and we go and get one in Mexico. Depending on where you're at and where you sample from, they're gonna look different. The plant microbiome is as diverse and equally as unique. And that means looking at the roots associated to the plant, the stem of the plant, or the leaves of the plant. So once again, if I do a swab of my forehead, a swab of my ankle, and a fecal sample, each part of my body is gonna have its unique microbiome. So on these graphs up here are some examples of what microbiomes look like on plants material. So this is called the edible microbiome. So in this study, the authors went and got different types of produce from a farmer's market and a grocery store. They brought it back, they washed it in water, They blotted it on paper, and they sequenced it. And guess what? It still had microorganisms. So when you go to the farmer's market and grocery store, and you buy produce, and you eat it raw, wash it or not, you are consuming microorganisms that are found in association with this. And this is the edible microbiome. And so I'm not going to try this. But you don't have to know a lot about what these graphs exactly mean, but looking at the pie charts, each of the color represents a different genus of bacteria, and the colors are kind of the abundance of each one. And so just looking at that, the green olives look completely different to the baby spinach, which look completely different to the tomatoes. And so by eating a variety of different produce and different edible microbiomes, can we have a positive effect on our gut microbiome? And there's a paper that came out just actually a couple months ago in September that asked these questions, and they went and they mined massive data sets. So, the power of sequencing, it's gotten so much cheaper, and there's so much data out there. And they started to ask these questions. Okay, so we know what microorganisms are going to be associated with some of these plants that we consume raw. Are they actually in the gut? Can we find them in the gut? And the answer is yes, they can. Do they persist in your gut? Yeah, they do. But what are they doing, and can they actually have a benefit in our gut? And yeah, some of these microbes have genes that produce these short-chain fatty acids and vitamins that are good for our overall health and well-being. And I highly encourage you, if you're interested in this topic, to read this paper. It is very cool. And one of the summaries, I hate wordy text, but I think this is important. This paper was not an agriculture paper. It was a bioinformatics, deep computational paper, but one of their big conclusions was that they challenged scientists to actually think about microorganisms from that soil, from that plant, and then what gets on your plate. What are you actually consuming, and how is that important for your overall well-being? Because microbes occurring in the soil, plants, and humans are interconnected. Therefore, it will be crucial to investigate how various plant cultivation methods affect fruit and vegetable-associated microbiota. So I want to change what I said before. The missing link between our diet and our overall health and well-being isn't just our gut microbiome. That was really, narrow-minded of me to say. And I want to change that to say it's microorganisms. It is not just what's in us. It is also important what is associated to the plants we're growing and how they're growing, and then what gets on our plate and in our stomach and in our colons. And microorganisms, they play a critical role from the field to the fork. From that raw material, thinking about how we grow the plants, what is the microbial foundation of seeds that we put into our soils? How are those microbes then getting into the seeds of the next generation or into those raw products? Microbes are really important in terms of food processing. We use them a lot for fermentation or for breaking down components and converting it into something else. Microorganisms are important in our own colon and for our own overall health, and so we have to think about taking care of them. And all of this ultimately will lead to hopefully better health outcomes. Thank you.
So I've never used one of these Slido things. But I'll read some of the questions from it. How can a grower evaluate whether or not their soil has a healthy microbiome? Shawn, as our soil scientist.
I'll repeat the question. How can a grower evaluate whether or not their soil has a healthy microbiome? Well, boy, that's a $10,000 question right there. We're pretty good with soil testing about telling you, all right, you're low in phosphorus, you're low in potassium, magnesium, whatever it might be. You're not managing your organic matter, and you got to do better at putting your, amendments together so that you're building organic matter, right? And if you were at the workshop on Thursday where we were talking about soil, I talked about soil testing. All that's pretty straightforward. But this is really complex stuff, right? And the microbiome in every soil is going to be different. So when you're talking about getting good information on any particular microbiome, it's going to have to be tailored to that soil, and you'll have to have an accounting of the management practices going into it. You know, it's complex. There are some soil testing options that will give you some information on the microbial community. Now, UK has regulatory services. They don't do microbial assessment, right? So when you send your soil sample in there, but there are some private options. For example, I think Waters Ag here in the state of Kentucky will do biological assessment. Now, it's not going to give you microbiome, but it'll give you some information on microbial activity and substrate utilization. In other words, the different types of materials in your soil, how able are the microorganisms able to break those down, and then that can be translated into maybe like nutrient cycling and nutrient uptake, you know. But we really don't have a good way of getting microbiome that's relatable to a producer right now. It's a real challenge. And I will say that even what Waters does, it's not microbiome, it's more microbial activity. It's expensive. Those soil tests are expensive. So, you know, it's an area where we stand a lot to grow and get, you know, as we figure some of this stuff out, this is kind of a burgeoning field. As we figure these things out, that is an area where a lot of people are interested in getting more information. It's a good question.
Okay, we'll next take this question about perennial plantings versus annuals. And Dr. Rodríguez López, can we get this microphone turned on, please? Okay, all right, forget it.
Well, the reality is that we really don't know. The majority of the information that we have about plant microbiomes is from annual plants, particularly from plants that are not crops, model plants. We know some about crops, but what I think you need to remember is that plants associate with very specific microbes. So the needs of one plant can be very different to the needs of another plant. And the other thing to remember is that, actually I'm gonna make this a question. Do you think that by putting a plant on the soil, we're increasing the diversity, the microbial diversity of the soil, or are we decreasing it? Anyone? We are increasing it? Well, no. The plants wants to, as I mentioned before, they're selfish. They want to associate with specific species that are beneficial or at least non-detrimental to them. So they are going to reduce the diversity. Studies that have been done looking at as you approach the rhizosphere of the plant, what we see is a decrease in diversity. So if you plant different species together, in a way they're going to be competing. But the reality is that the short answer is we don't know. There is not much information about perennial species and microbiomes. I'm trying to study this with grapevines as a model, for example.
Shawn, we're going to talk about mycorrhizal structures, if you want to talk a little bit about that. But I think, putting in food stocks or things that fungi, you can derive the fungal components of soil. Shawn, I think you know about this. We looked at some of this kind of stuff. Things that fungi break down preferentially, wood, things like that. We can use high carbon materials in the soil versus like a high nitrogen material that can affect bacterial fungal ratios in the soil. So we can, in our farming practice, do that. And Carlos, what you said there just is making me think. I think in a lot of people here that do vegetables, diversity in your system. If you're doing lots of different vegetables, you're using multi-species cover crops, you can affect the diversity in the system versus monocropping. So we can, through our farming practice, get more balance, more diversity, more abundance in the plant communities that we grow in our systems. But you're seeing a theme here, and I said this at the beginning. we're looking at a continuum here from farming practice to human health, mediated through this kind of microbial lens. And there are some things that we do know. A lot's been known from a soil standpoint for a long time, and there's an increasing amount of evidence about the way that microbes interact with plants. But like in Kendall's research, it's right at the cutting edge, and it's the same thing with plant microbiome interactions. A lot of this stuff is unknown, and hopefully one day there will be a better, more cost-effective ways for farmers to look at the microbial community, the microbiota in their soils. But right now, it's right at what we know and what we're capable of looking at. So, it's an exciting field, because when we think about the human health and what we're seeing increasingly in terms of how the human microbiome affects our health, it's amazing. And to think that our farming practice could be affecting that, it really is mind-blowing to justify a lot of the things that are at the core of organic farming, not just from your soil or productivity in terms of whatever dollars in your crops, but how it goes all the way through to the human health. I think it gives us more justification for being organic farmers, and I think it is clear that organic farms, plants, and systems have increased microbial community health, abundance, diversity, all of those kinds of things. That has been shown. So I think it's more fodder for why we can justify a lot of the practices that are at the core of organic farming.
Can I say something very quickly about
Please.
diversity? I know that diversity is the holy grail in a way, but I don't think many of you would be absolutely happy if I came with a bunch of pathogens and dropped them in your farm. I would be increasing diversity, but I wouldn't be increasing a good diversity. So diversity is good if it's the good diversity.
Yeah. And this second point, maybe we have time for this one last one. Is there any evidence that microbiome in soils or plants and organic systems is more diverse, robust, or healthier compared to the microbiomes in conventional? And the answer to that is yes. We published a paper in 2015 that looked at 4 crops grown conventionally and organically: pepper, melon, corn, and tomato, and we compared soils that have been farmed conventionally. This is on the Horticulture Research Farm in Lexington. For like 30 years versus same soil, Mari silt loam soil that was farmed organically for 10 years. And we extracted out microbes from these plants. We cultured those microbes, then we sequenced the 16S ribosomal RNA, and we characterized those, and we showed that organic practices increased diversity, abundance, richness, all of these factors. And it was, as Carlos said, it was genetically based, different plants associated with different microbes. It was component of the plant-based. Roots had different microbiome than in the stems, than in the leaves, and then in the seeds, which had the highest level to the lowest level in that spectrum right there, and at the different life stages of the plant. So the reason this is so complicated is there's so many factors that control this ecosystem. But that was published in 2015, and there's an increasingly amount of data out there to show this at we use culturable techniques, or bacteria that we could culture, which is just a small percentage of the microbial community. There's a lot more advanced techniques that people are using now and having a much clearer picture on this. But the answer, I think, is yes on that one. So thank you to the speakers.